Languages and language translators in DNA, RNA, and protein systems point to God. The presence of languages and language-to-language translators in DNA, RNA, and protein systems points to intelligent design by God. Languages are always the product of or derivative of an intelligent mind or minds. It is incoherent to claim that languages arose by random chance or by natural law. Language-to-language translators are always the product of or derivative of an intelligent mind or minds. It is incoherent to claim that language-to-language translators arose by random chance or by natural law. Therefore, the presence of languages and language-to-language translators in DNA, RNA, and protein systems points to intelligent design by God. Introduction. The field of molecular biology has revealed that life is driven by intricate systems of information processing, much like the digital communication systems we use in technology today. At the core of this biological information processing are languages and language-to-language translation mechanisms embedded in DNA, RNA, and protein systems. These systems do more than just store genetic information. They interpret, translate, and execute instructions, enabling the complex processes of life to occur. This video explores the concept of languages within biological systems and the translators that convert information from one language to another, underscoring the complexity and sophistication inherent in life at the molecular level. The language of DNA, the genetic code. DNA, or deoxyribonucleic acid, is the molecule that contains the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms. The genetic code embedded in DNA is a language because it uses a specific set of symbols, nucleotides, arranged in sequences to convey information. 1. Nucleotide sequences as letters. DNA is composed of 4 nucleotides: adenine, A, thymine, T, cytosine, C, and guanine, G, which can be thought of as the letters of a biological alphabet. These nucleotides are arranged in specific sequences to form genes, much like letters form words and sentences in human languages. 2. Codons as words. A codon is a sequence of 3 nucleotides that corresponds to a specific amino acid or a start or stop signal in protein synthesis. These codons can be likened to words in a language, where each word has a distinct meaning and function. 3. Genes as sentences. Genes are sequences of codons that encode the instructions for building proteins, the molecular machines, and building blocks of life. In this sense, genes function as sentences, providing complete instructions that can be read and executed by the cell. 4. The genetic code. The genetic code is universal across almost all organisms, meaning the same codon sequences produce the same amino acids in different organisms. This code is the language that DNA uses to store and transmit genetic information. The language of RNA, transcribing DNA's message. RNA, or ribonucleic acid, plays a crucial role in translating the genetic information stored in DNA into functional proteins. RNA acts as an intermediary, transcribing the language of DNA and helping to interpret it within the context of protein synthesis. 1. mRNA as a messenger. Messenger RNA, mRNA, is synthesized from DNA in a process called transcription. During this process, the sequence of nucleotides in a gene is copied into an mRNA strand. This mRNA strand carries the genetic message from the DNA in the nucleus to the ribosomes in the cytoplasm, where proteins are synthesized. 2. tRNA as a translator. Transfer RNA, tRNA, is the molecule that translates the language of nucleotides into the language of amino acids. Each tRNA molecule has an anticodon that pairs with a specific codon on the mRNA and an attached amino acid corresponding to that codon. This translation process ensures that the correct amino acids are assembled in the correct order to form a protein. 3. rRNA as the machinery. Ribosomal RNA, rRNA, is a component of the ribosome, the molecular machine that facilitates the translation of mRNA into a protein. The ribosome reads the mRNA sequence and uses tRNA to assemble the amino acids in the correct sequence, producing a functional protein. Protein systems, the executed language of life. Proteins are the end products of the genetic information encoded in DNA and transcribed and translated by RNA. They carry out the instructions encoded in the genetic language, functioning as enzymes, structural components, and signaling molecules within the cell. 1. Amino acids as words in protein language. Amino acids are the building blocks of proteins. Just as codons and mRNA correspond to specific amino acids, the sequence of amino acids in a protein determines its structure and function. The language of proteins is thus a sequence of amino acids, each contributing to the protein's overall meaning or function. 2. Protein folding and function. Once synthesized, a protein folds into a specific three-dimensional shape, determined by the sequence of amino acids. This folding is crucial because the shape of a protein determines its function. The process by which a protein achieves its final form is analogous to the way a sentence's meaning is understood through its syntax and grammar. Language-to-language translators in biological systems. The processes of transcription and translation in DNA, RNA, and protein synthesis can be viewed as a form of language translation, where the genetic code is converted into functional proteins. 1. Transcription, DNA to RNA. During transcription, the language of DNA is transcribed into the language of RNA. This process is akin to translating text from one language to another while retaining the original message's meaning. The transcription machinery ensures that the nucleotide sequence in DNA is accurately copied into mRNA. 2. Translation, RNA to protein. Translation involves converting the language of RNA into the language of proteins. The ribosome reads the mRNA sequence and, with the help of tRNA, assembles the corresponding amino acids into a protein. This process is a direct analogy to translating a sentence from one language into another, ensuring that the original instructions are faithfully executed in a new form. 3. The role of ribosomes. Ribosomes are the molecular machines that facilitate this translation process. They ensure that the mRNA is read correctly and that the amino acids are assembled in the precise order dictated by the genetic code. The ribosome itself can be thought of as a sophisticated translation device, capable of interpreting and executing the instructions encoded in the genetic language. Conclusion. The implications of biological languages, the presence of languages and language-to-language translators in DNA, RNA, and protein systems, reveals a level of complexity and precision that is strikingly similar to, but vastly superior to, human-engineered communication systems. These biological languages are not only capable of storing and transmitting information, but also of translating it into functional outcomes that sustain life. The existence of such sophisticated systems raises profound questions about the origins of life and the mechanisms that drive biological processes. The complexity of these language systems, coupled with their precision and universality, shows a design and intentionality that falsify purely naturalistic explanations for the origin of life. Instead, these point an intelligent cause behind the creation of these systems, capable of encoding and interpreting the languages of life. In summary, the presence of languages and translators in DNA, RNA, and protein systems provides a powerful framework for understanding the intricacies of biological information processing. This highlights the remarkable capabilities of life at the molecular level and underscores the sophistication inherent in the design of living organisms. Conclusion 1. The presence of languages and language-to-language translators in DNA, RNA, and protein systems points to intelligent design by God. 2. Languages are always the product of, or derivative of, an intelligent mind or minds. It is incoherent to claim that languages arose by random chance or by natural law. 3. Language-to-language translators are always the product of or derivative of an intelligent mind or minds. It is incoherent to claim that language-to-language translators arose by random chance or by natural law. 4. Therefore, the presence of languages and language-to-language translators in DNA, RNA, and protein systems points to intelligent design by God. 5. The presence of languages and language-to-language translators in DNA, RNA, and protein systems proves intelligent design by God, beyond a reasonable doubt. Thanks for watching. Please like, share, and subscribe, and comment below if you agree or would like to add your thoughts.